All right, engineers, in this video we're going to go into a lot of detail on the electron transport chain. Okay, so where does the electron transport chain occur? Well, if you imagine here, let's say we take a big deep look into the mitochondria. This part right here, this line right here, this whole thing right here is the outer membrane of the mitochondria. So this is the outer membrane of the mitochondria. Then there is the inner membrane of the mitochondria, which is going to have a lot of nice folding. You see how it's really folded? That folding of the actual mitochondria is called the cristae of the mitochondria. Okay, so how do you say that? It's called the cristae. So again, here it is. It's the cristae of the mitochondria. And it's the inner mitochondrial membrane. So again, you get the outer membrane of the mitochondria, and then you have the inner membrane of the mitochondria, which is going to be folded, so they call it the cristae. And then you have this space. You see this space right here from the inner mitochondrial membrane to the outer mitochondrial membrane? They call that the intermembrane space. Then inside of the mitochondrial membrane, the inner mitochondrial membrane, this is going to be what's called the mitochondrial matrix. This is where a lot of the Krebs cycle activity is occurring and a lot of stuff that you've seen in a lot of these reactions with the conversion of specifically the pyruvate to acetyl-CoA and then the Krebs cycle. So again, one more time, this is the outer mitochondrial membrane, which is close to the cytosol. So this is where the cytosol of the cell would be. Then you have the intermembrane space. Then you have this inner mitochondrial membrane, which is folded, so it actually is called cristae. And then you have the inner fluid of the mitochondria, which is called the mitochondrial matrix. Specifically, the electron transport chain is occurring on the inner mitochondrial membrane. And we're going to talk about each component of that in very great detail. So let's go ahead and get started. So how does all of this actually occur? Well, if you remember, we had to start with the glycolysis. So we took glucose, which was that 6-carbon unit, oxidized it, generated some NADHs and a little bit of ATP, 2 ATP net, and made it and converted it into 2 of these suckers right here. So 2 of these puppies. What are these 3-carbon units right here? This is called pyruvate. Pyruvate. This black marker looks good. So glucose to 2 pyruvate molecules, right? And this is going to be splitting a 6-carbon molecule into 2 3-carbon molecules. And you're generating these 2 NADHs, which will take them to the electron transport chain. We'll have another part of this video where we'll take the NADHs and show how they get into the actual mitochondria, because there's a special mechanism for that. Okay, then that pyruvate, if there is oxygen present, the pyruvate gets pushed into the actual mitochondrial matrix through specific transporters, and then it gets acted on by the pyruvate dehydrogenase enzyme, if you guys remember that complex. And then it converts it into acetyl-CoA, which is this 2-carbon unit. So again, right here is your pyruvate. You guys should know all of this already. And then this is going to be your acetyl-CoA, right, which was generated from that pyruvate dehydrogenase enzyme. What was the significance of that? You generated 2 NADHs from that reaction. And then if you remember, we're not going to go each step here because we've already done it. You know that you'll go through the Krebs cycle. And through specific sequences of the Krebs cycle, you generate how many NADHs? You generate 2, 4, 6 NADHs. And then also you generate 2 FADH₂s. So this is the significant part right here. How many FADH₂s did I generate? 2. And how many NADHs did I generate total? Let's show that in one whole go here out of this. Out of all of this, I generated a total of 6 NADHs from the actual Krebs cycle. That doesn't include the 2 NADHs from the transition step and the 2 NADHs from glycolysis. Now, these NADHs and what else? These FADH₂s that I generated, the 2 FADH₂s here, they're going to be very special because they're going to take those hydrides. If you guys remember, they're called hydride ions. What's a hydride ion, if you guys remember quickly? It's just specifically a proton with 2 electrons, right? That's all it is. That's all hydride is. It's just a proton with 2 electrons. These guys are carrying that. Now, these 2 guys are our electron shuttles. What they're going to do is they're going to take those electrons and take it to this actual electron transport chain. So let's focus in on specifically how NADH is doing it first, and then we'll do FADH₂. Okay, so here we're going to take our NADH. So we're going to take one NADH and see how it's reacting right here. So what happens is you have this NADH. This NADH is going to come into this action, it's going to react with this first part of the electron transport chain. And what's going to happen is it's going to go in as NADH and it's going to come out as NAD positive. What happened in this reaction that NADH got oxidized into NAD positive? You know, inside of here, there's actually going to be specifically a flavin mononucleotide. So here we're going to have this thing called a flavin mononucleotide. That flavin mononucleotide is going to pick up those electrons off of the NADH. And then it's going to get converted into flavin mononucleotide, which is carrying the actual electrons. So this is going to be the flavin mononucleotide carrying the electrons. And then it can also pass it on to iron sulfur clusters. But specifically, what's happening at this first step here? NADH is getting oxidized to NAD positive, and then flavin mononucleotide is getting reduced into flavin mononucleotide with the electrons. And then what happens? This flavin mononucleotide will react with this guy over here. This guy over here is going to be called coenzyme Q. So look what we have here. I have coenzyme Q. Coenzyme Q is going to come and react with that FMNH2. Look what happens. He's actually going to pick up the electrons from this FMNH2. When he picks up the electrons from an FMNH2, he gets converted into coenzyme Q, and specifically they call it H2. Okay, so again, what happened here in this first part? NADH dropped off its electrons to the FMN, who got converted into FMNH2, which is the reduced form of flavin mononucleotide. That flavin mononucleotide, he passes the electrons onto coenzyme Q, which gives you the reduced form of coenzyme Q. And then he'll take these actual electrons and pass them on to the next step. Before we do that, now let's see how FADH2 is doing this. Okay, because now we got to look at how exactly FADH2 is performing this function. Okay, FADH2 is going to come to this one right here. So let's show FADH2 coming up over here. FADH2 is going to come onto this specific component of the electron transport chain. And what it's going to do is it's going to drop off his electrons. Okay? He's going to drop off his electrons. And when he drops off his electrons, what would he come out as? He would come out as FAD. So he gets oxidized. When he gets oxidized, who gets reduced? If you remember again, over here I'm going to have the flavin mononucleotide. He's going to pick up those electrons and get converted into flavin mononucleotide, which is in the reduced form with the actual electrons. Then what happens? Look what else I have over here. You know this guy right here, this Q, this coenzyme Q, you know he's a mobile molecule. He's also not made of protein either. He's one of the only ones throughout this entire electron transport chain that's not made of protein. He is actually mobile along the membrane, so he can run ahead. And as he runs ahead, he gets over here. And now look at him. Coenzyme Q right here, he's like, hey, FMNH2, give me those electrons, I'll take care of them. And he gets converted into the reduced form of coenzyme Q, which is QH2. Okay? So what has happened so far with the FADH2s and NADHs? NADH dropped his electrons off onto this first complex. You know that's what we call it? We call this specifically, you know what we call this first one? It has two names. I can either call this complex 1, or I can call this NADH dehydrogenase. Because what did I tell you guys? Anytime you have NADH being dealt with, what do you know is going to be there? A dehydrogenase enzyme. So there's going to be an NADH dehydrogenase, or you can also call this one complex 1. Then, where did these electrons go from complex 1, or the NADH dehydrogenase? They get passed on to coenzyme Q, and then coenzyme Q gets reduced. So this is actually going to be specifically, what's this molecule here called? This guy right here is called coenzyme Q. And sometimes you might even see it written as coenzyme Q10. Okay? Then, this coenzyme Q, where is he going to pass those electrons? We didn't talk about that yet. We're almost getting there. Before we do that, let's see what happened with the FADH2. FADH2 dropped his electrons off onto this second complex, if you will. So what is this guy here called? This is actually going to have two names again. I can call this one specifically, I'm going to call this one Succinate Dehydrogenase. Or they can also call it specifically complex 2. Now, complex 2 or succinate dehydrogenase is going to do what? It's going to take the FADH2 and convert it into FAD. Doesn't this enzyme sound familiar? Do you remember this enzyme, guys? It was actually occurring here in the Krebs cycle. Remember when you were taking succinate and you were converting it into fumarate? What was it doing? It was taking FAD and converting it into FADH2. But remember I told you it's not a heavily regulated step. So what does that mean? It's reversible. So this is the enzyme that's actually going to be involved in the Krebs cycle as well as in the electron transport chain. Now, before I move on, I want to explain something with respect to complex 1 or NADH dehydrogenase. But I have to do it with a graph. So let's say over here I have a graph. And on the y-axis, it's measuring energy. And on the actual x-axis, it's like the reaction progress. Okay. Let's say I take this first NADH dehydrogenase. And he's sitting here at lower energy. And I take the electrons from that NADH and I pass it on to him. If I pass that on to him, he's going to go to high energies. Who is this one? This is complex 1, or the NADH dehydrogenase enzyme, right? The complex. When this actual enzyme complex 1 goes to high energy levels, it doesn't prefer it. It doesn't like to be at high energy levels. He doesn't like that. No one likes to be at high energy levels. So what he decides to do is he decides, I don't want these electrons. I'm going to pass these electrons on to someone else. And when he does that, he loses the energy, and he goes back down to low energy levels. Okay, well, when he goes down to low energy levels from passing what? Passing the electrons away, what happens? He has a special type of pore. So let's say here is a component of this actual enzyme complex. Look over here. It has a pore. And on this pore, it can do something really special. What was NADH again? It was consisting of hydrides. What's a hydride? It's a proton and 2 electrons. All we did was pass electrons onto this complex 1. Where'd the protons go? These protons are going to get pumped out. Okay? And it's going to get pumped from the actual mitochondrial matrix into the intermembrane space. And what allows for this? Whenever the electrons from complex 1 is getting passed on to coenzyme Q. Now, coenzyme Q takes these electrons and he'll pass it on to someone else, which we haven't talked to yet. But what about complex 2? Let's say here I have complex 2. Complex 2 is going to get these electrons from FADH2. He's going to go to high energy levels. When he goes to high energy levels, he doesn't like it, so he passes it on to the other coenzyme Q, and it goes back down to low energy levels. But look, he doesn't have a pore. He doesn't have a protein component that will pump the protons out. So no protons are getting pumped out through the actual mitochondrial matrix and into the intermembrane space at complex 2 or succinate dehydrogenase. Only complex 1. Okay, now it's time that we explain this next part. So this QH2, that coenzyme QH2, which is in the reduced form, what's he going to do? He's going to take those electrons and he's going to pass those electrons onto this other coenzyme Q here. So he's going to pass these electrons onto that coenzyme Q and help to form another QH2. Who else is doing that? This FADH2 is giving it to FMN, who's getting converted into FMNH2, who's also helping to convert coenzyme Q into the reduced form of coenzyme Q. And again, what is this molecule here called? This right here is specifically called coenzyme Q10, right? Or ubiquinone. They even call it ubiquinone. Now, what happens with this ubiquinone? Well, now he's going to take those electrons and pass it on to the next guy. This is complex 3. So now we're going to go into the next one, which is going to be specifically complex 3. But you know they have another name for complex 3? So this is complex 3. But they also call complex 3 something else. They also call complex 3 a weird name. It's called coenzyme Q, Cytochrome B, oxidative. Okay, so again, what is the name of this? Coenzyme Q, cytochrome B, oxidative. That is its name, or you can even call it cytochrome BC1 complex. Or you can just call it complex 3. It's up to you guys. All right, but anyway, what's happening here? You have this cytochrome, and you know cytochromes are interesting because they're basically kind of like heme groups. They have a porphyrin component of them, which is like a pigment, as well as having some type of, usually an atom or mineral, or specifically a metal in the center component of it. In this case, it's iron. So let's say I have cytochrome B, and I draw here cytochrome B. And the cytochrome B specifically has iron here, and it can have an iron here. Now, what happens is this coenzyme Q, H2, right? It's going to pass these electrons onto the cytochrome B. When it passes the electrons onto the cytochrome B, who is it specifically going to pass it on? Well, you know iron can exist in 2 states, right? So let's say that he passes these electrons over here. And when he does that, this guy gets converted right back into his original state because he's giving those electrons away. He's going to accept these electrons. When he accepts these electrons, he's going to get turned into a different oxidation state. You know what state he's going to be in here? It's going to be +3. If he gains an electron, one at a time, right? What would it become? +2. So specifically what's happening is that coenzyme QH2 is passing this electron on to Fe+3. And Fe+3 is getting converted into Fe+2 because he's accepting that electron. Now, same thing like before. Let's say I have this graph here. I keep extending my graph. If I keep extending my graph now, what's this enzyme complex 3 going to be at before it accepts the electrons? At low energy. But then it accepts that electron. And when it accepts those electrons, it goes to high energy. He doesn't like it. What is he going to do? He's going to pass the electrons onto the next guy, which is going to bring him down into low energy. You know what's special about this guy, just like complex 1? He has another channel off the side. And off to the side of this guy, he's got this little pore. And you know what this pore is going to be doing? It's going to be pumping protons out here into the intermembrane space from the mitochondrial matrix. Because you know there's going to be protons which are going to be concentrated in this area, right? The concentration in here, you're going to have a lot of protons in here too. Usually this concentration in this area is usually around 10 to the -9, right? And specifically this might be in millimolarity, right? So you're going to have some type of concentration out in here, and usually before these protons are getting pumped out into this area, it's also about 10 to the -9 millimolarity. So usually they're pretty much equal before you start pumping tons of protons out. We'll see how it changes. But it's pumping these protons out into the actual intermembrane space. At what point? At complex 3 and at complex 1. Or you can also call complex 3 cytochrome Q, specifically coenzyme Q, cytochrome B, oxioreductase. Then what happens? This guy right here is going to pass his electrons on to the next guy. Okay, who is this next guy? This guy right here is specifically going to be called Cytochrome C. So this is called Cytochrome C. So they call it Cytochrome C. So what Cytochrome C is consisting of is still the same. It's going to have that heme group in it, right? So here, let's say you have your Cytochrome C. And again, it's going to have iron in it. And it might even have copper as well incorporated into it also. But again, what's happening? This iron passes electrons onto this Fe2+. This Fe2+ is at high energy states. He doesn't want to have those electrons anymore. What is he going to do? He's going to take and pass those electrons onto this guy. So now look what happens. These electrons are going to get passed over onto this guy, and he's going to get regenerated back into Fe3+. When he does that, this guy is now going to do what? Well, specifically, let's say that we do the same thing. Let's say that this iron right here reacts with that and picks up those electrons. If he picks up those electrons, what state should he be in? He should be in the +3 state. When he accepts those electrons, he should now go into the +2 state. And again, you can have copper as a component of this cytochrome too. Then what happens? Then we go on to the last one. At the last one, we have it called as complex 4. Or, like always, it can also have another name. They also call this Cytochrome Oxidase. And you'll see why they call it oxidase. This is a very, very important complex. Okay, now, same thing. It's going to become redundant here. And I know This is going to be specifically, this cytochrome oxidase is actually consisting of cytochromes, but specifically cytochrome A and also A3. So cytochrome A plus A3. So it consists of these two cytochromes in its actual enzyme complex. And once again, this cytochrome A plus A3 is going to be consisting of iron. And what's happening here? This iron is in the +2 state. So if he's at the +2 state, he's not going to like that. He's at high energy status. He doesn't want to be at high energy status. He's going to take those electrons and pass it on to the next guy. It's just like a game of hot potato, right? I don't want it. Who am I? I'm enzyme complex 1. I'm going to give it to the next guy, coenzyme Q. Coenzyme Q doesn't want it. He gives it to another coenzyme Q. 2 doesn't want it. He gives it to coenzyme Q. Q gives it to who? Cytochrome B. Cytochrome B gives it to cytochrome C, and cytochrome C gives it to this actual cytochrome oxidase. And it's just a game of hot potato all the way down. Now, when this guy passes his electrons back over here, because now look, he's going to come from here, and he's going to pass these electrons back over to this guy. What do you think this guy's going to do? Same thing. He's going to pick and accept those electrons. When this iron, which is going to be, again, redundancy, +3, he goes back into the +2 state. He accepts those electrons. Then what happens? Something very, very interesting happens. You know, in this area, I'm going to have oxygen. So I'll have oxygen out here. So let's say here I have some oxygen. And specifically, it's the atomic oxygen. So if I put 1/2 of an oxygen, that's just specifically atomic oxygen. That 1/2 of an oxygen is going to react with this iron in the +3 state and this iron in the +2 state, specifically the +2 state one. So as this iron in the +2 state is going back to the +3 state, he's giving his electrons to who? He's giving it to this oxygen. This oxygen is then going to pick up those electrons from that +2 iron. You know what else is going to get combined in there? 2 protons. So now these 2 protons are going to get combined with the half of an oxygen. So you're going to have 1/2 of an oxygen taking up these electrons from the +2 iron in the cytochrome A plus A3 going back to +3. He's going to accept those electrons, combine with 2 protons, and make water. Oh, man, it's beautiful. So what happened here then? I took half of an oxygen, 2 H pluses, and I accepted those electrons. Do those electrons go anywhere else? No. That means that this oxygen is the final electron acceptor. So it went hot potato, hot potato, hot potato, and then gave it to oxygen. And he just accepted it, and he's like, okay, I'll take the potato. Right? So he is going to be the last one of the electron transport chain accepting those electrons. So now, before we go back over here and review this, one more thing happens. Just like this guy, let's come back to this graph. This complex 4 is at low energy levels. But then what happens? When this complex 4 accepts the electrons from this cytochrome C, right? The +2 iron is giving the electrons to the cytochrome A plus A3. He's accepting those electrons and going to high energy levels. Does any molecule prefer high energy levels? No. So what does he do? He gives those electrons to who? To the oxygen, and then the protons to make water. When he passes those electrons away, his energy levels go back down. That energy is harvested to do what? Look what happens here. What do you think I have right here into this membrane? I have a little channel. And guess what I'm going to pump out of this sucker right here? I'm going to pump out a proton. So I'm going to take a proton and I'm going to pump that proton where? From the intermembrane space into the, from the mitochondrial matrix into the intermembrane space. So now, let's review from beginning to end what's actually happening. So where did we start? We started at complex 1, or again, NADH dehydrogenase. The NADH is dropping the electrons off to this complex 1, specifically the flavin mononucleotide, and they're also, just remember, I'm going to write it down. Technically, this flavin mononucleotide is passing the electrons before it goes to coenzyme Q. It's passing it to a lot of iron-sulfur cluster proteins. It's not significant in this concept, just know that it's passing it to a lot of iron-sulfur cluster proteins. Anyway, once this NADH passes electrons, it goes to NAD positive, and then this FMN gets converted into the reduced form of FNN. And technically, this FMN, or reduced form, is going to pass it on to a bunch of iron-sulfur cluster proteins, which is then going to pass those electrons on to who? Specifically, it's going to pass it on to coenzyme Q. Coenzyme Q is going to be a mobile, specifically what? It's a mobile electron transport chain component, but it's not made of protein. The coenzyme Q is going to accept those electrons and get converted into the reduced form of coenzyme Q. And he'll pass those electrons on to another non-reduced form of coenzyme Q, to make the reduced form of coenzyme Q, because he is able to be mobile. He can be throughout this actual inner mitochondrial membrane. At the same time that's happening, FADH₂ is also being converted into FAD. He's getting oxidized. Who's picking up those electrons? The flavin mononucleotide and the enzyme component of succinate dehydrogenase. And then it's getting converted into the reduced form of flavin mononucleotide. Okay, and this also consists of some different types of iron-sulfur cluster proteins. But then what happens to this reduced form of flavin mononucleotide? The coenzyme Q is going to pick up those electrons and get converted into the reduced form of coenzyme Q. Also remember, at complex 1, what was happening when these electrons were getting passed over? It was pumping a proton out. Then these electrons from the actual complex 2, where the succinate dehydrogenase gets passed on to, specifically coenzyme Q here, also coming from the coenzyme Q before, this coenzyme Q is going to react with Q, cytochrome B oxioreductase, or you can call it cytochrome BC1 complex, or you can just call it complex 3 specifically. What's happening here? This cytochrome B is consisting of iron, and this iron components here are going to be accepting the electrons. Which one? Iron in the +3 state is going to accept the electrons from the coenzyme Q and go into the +2 state. Then it's going to take those electrons and pass it on to another iron who's going to be in the +3 state, so they can go into the +2 state in the component of Cytochrome C. But when it passes those electrons over to the Cytochrome C, he goes from high energy to low energy and pumps out a proton from the mitochondrial matrix into the intermembrane space. Then the Cytochrome C is going to do what? When it accepts those electrons, it goes from the +3 iron state to the +2 iron state. And then again, it goes to high energy, doesn't want it, passes electrons onto cytochrome oxidase, or enzyme complex 4, which is consisting of cytochrome A plus A3. Then what happens? This is also consisting of iron, and he accepts those electrons from the cytochrome C and goes from the +3 state to the +2 state. Doesn't want those electrons, too high energy. What does he do? He passes electrons onto oxygen, who's the final electron acceptor, who will then combine with 2 protons to make water. But when he goes from the +2 back to the +3, back to low energy levels, it opens up this port to push a proton out. So again, at what points here are we actually pushing the protons out? Specifically, we're pumping a proton out at complex 1. We're not pumping a proton out at complex 2. We're pumping a proton out at complex 3. And we're pumping a proton out at complex 4. And this is due, again, to whenever it's from high energy transitioning to lower energy. Now, as all of this is happening, what's happening to the proton concentration out here now? Now we can go in and explain to this next part, at least for a second. Now the proton concentration out here is going to be much higher than the proton concentration in the mitochondrial matrix, because we're pumping tons of protons out here. This proton concentration can go up until about 10 to the -7 millimolar. Whereas what was it compared in the mitochondrial matrix? It was about 10 to the -9 millimolar. So now there's actually a 2-fold increase in the protons out here in the intermembrane space. And this establishes what's called a concentration gradient. And we'll talk about that in a second. So now, what's the problem though? We're not done yet. And you want to know why? We took care of the NADHs from the Krebs cycle, these 6 NADHs, and we took care of the 2 NADHs from the transition step. And we took care of the 2 FADH₂s from the Krebs cycle. But what NADHs have we not accounted for yet? These suckers right here. Now we have to figure out, because here's the problem. NADH can't just cross the mitochondrial membrane. It has to have a special, obviously complex way to get in and then drop those electrons off. That's what we're going to do in the next video. So in the next video, we're just going to continue on with the electron transport chain, but talking specifically about how these NADHs are getting into the mitochondria and doing these functions.